Okay, so the next topic that we're going to talk about is cell division. And we're going to start off first with mitosis. So, if we think about this process of mitosis, the cell cycle is really important. It's what allows our cells to divide. It's an important process because it is through the division of cells that organisms are able to grow, to repair, and to reproduce. Now, when we're talking about cell division, what we can do really depends on whether we're talking about a unicellular or multicellular organism. When it's just a single cell, division of that cell results in an entirely new organism, like we see of this amoeba right over here. You had one, you separate, and now you have two. For multicellular organisms like us, though, cell division is important in making reproductive cells so that we can also reproduce. But cell division is also the process that allows for us to grow larger and also to replace and repair damaged cells and tissue. So when we're thinking about what we can do, a unicellular organism, it's just to divide and reproduce. Multicellular organisms, our cell division involves both reproduction, growth, and repair. When we think about cell division, it's important, but cell division is only one part of the cell cycle, albeit a very important part. We'll get into the rest of the cell cycle in a minute, but first let's look at the cell division part. In eukaryotic cells, there are two different types of cell division, meiosis and mitosis. Meiosis is a special case of cell division. It's important because it produces cells that are genetically different from the original cell, and it is the form of cell division used to reproduce or to reproduce sexually to produce gametes. In mitosis, it's more of the regular cell division. It produces cells that are identical or carbon copies to the original cell, and it is the type of division necessary for asexual reproduction, growth, and also tissue repair. So if we think about it in terms of the life cycle, these two different forms of cell division have their very own place and role. But when we talk about nearly all sexually reproducing life cycles, you can see that both forms of cell division are needed. So here we have the human life cycle. We have the adults, and the adults produce gametes, that's your egg and your sperm, via the process of meiosis. This is the only place in the life cycle of animals where meiosis occurs. These gametes then fuse at fertilization to produce a zygote. From this point forward, right, because that zygote is a single cell, but from this point on, the organism uses only mitosis, and it uses mitosis to grow, to go from being one cell to two to four, four to eight, and so on until the adult size is reached. In addition to growth, though, our cells undergo mitosis for repair and replacement throughout our lives. Okay, so when we think about the life cycle of most animals, meiosis is a special case. It occurs in one time and in one location. Mitosis is what is predominantly happening throughout the life cycle. So meiosis, remember, is involved in making our sex cells, which are also referred to as our gametes, or what we use to reproduce. So after meiosis, we have production of 4 non-identical daughter cells, which are called gametes. In humans, these are the egg and the sperm. The very other important part of meiosis is that those daughter cells that are produced have half the number of chromosomes that the parent did. This is referred to as them being haploid. They have half the normal complement of chromosomes. And this is really very important. So we'll talk about it much more when we get into meiosis. Because when the egg and the sperm fuse at fertilization, the chromosomes from each of those gametes are combined to get back to our total number of chromosomes, which in humans is 46. If it wasn't reduced, each time fertilization occurred, we would be increasing the number of chromosomes, and that would result in mutation or in problems, genetically speaking, over time. When we think about mitosis, on the other hand, mitosis occurs in our cells that are referred to as somatic. So somatic cells are every cell in your human body aside from your germ cells. These are the non-reproductive cells. After mitosis, what we end up with is 2 daughter cells, and those daughter cells are genetically identical to the parent. Because these daughter cells are genetically identical, they must also have the same amount of chromosomal material as the parent did. So they are said to be diploid because they have 2 copies of each chromosome, or a full complement. Now, when we are talking about chromosomes and DNA molecules, each eukaryote has a characteristic number. We used to think that larger organisms or more complex organisms would require more DNA, but we know that's just not the case. So if we think about humans, we know we have 46. These cute little hedgehogs have 90. These elephants here have 56. Do you know which species has the smallest number of chromosomes? It happens to be this critter right here. This is a jackjumper ant. And this individual, he only has a single chromosome. And the reason he only has a single chromosome is because in many insects, their reproductive life cycle is called haplodiploidy. And the way that gender is determined is by whether or not the egg is fertilized. If the egg is fertilized, it's a female. If the egg is not fertilized, it's a male. And so all of the males of that species are haploid. They only have 1 copy of every chromosome. So whereas, we have those 2 copies of chromosomes, one came from mom and one came from dad, they only have 1 copy of every chromosome that came from mom. So the smallest number of chromosomes an organism can have is 1. On the opposite side, the organism with the largest number of chromosomes is this one right here. This is a fern, and it's called adder's tongue. It has 1,440 chromosomes. Now I want you to think about this, 1,440 chromosomes that are shoved, compacted down into a teeny tiny little cell that you can't see without a microscope. If you remember when you looked at your cheek cells have 46 chromosomes. If we would take all of our chromosome material and put it end to end, we'd have about 3 meters of DNA. 3 meters of DNA. And if we think about that, that's scrunched down into a structure that you can't see without a microscope. And this plant has 1,440 chromosomes per nucleus. That's impressive. Okay, so when we think about this whole process, before we can go through cell division, the first thing we need to do is we need to be able to replicate our DNA. And so when we replicate our DNA, we go through a variety of different stages. This is the wonderful thing about mitosis and meiosis, is that when we talk about DNA is called something different depending upon what state of compaction it is in. Right? So if we think about the overall organic molecule, that's DNA, right? That's our nucleic acid. When we start to compact our DNA down, our nucleic acid down, we wrap it around these proteins called histones, which is what you see happening here, and we call it chromatin. The chromatin is, think of it kind of like a big pile of spaghetti. You're compacting it down a little bit, but it's still hanging out all over the place. Okay? When we compact it down even more, we call it a chromatid. Okay? So when we're getting ready to replicate our cells, we need to pack down our DNA. And the reason for this is because if we left it in that chromatin form, where it was a big giant pile of spaghetti, and then we tried to separate it apart, you would have a much greater chance of causing breakage of your DNA. And so when we compact it down, we're packaging it up for separation. Okay? It's kind of like if any of you would admit to being knitters, right? When you are knitting something and you're actively using it, you have to have it all spread out so that it's easy to thread around the needles. But when you're getting ready to go somewhere, if you leave it like that, it'll get all knotted up. So you take your thread and you wrap it around the needles and you compact it up all nice and neat so that you can travel with it. And that's basically what your cell is doing. Okay. So this is our DNA all packaged up, but what we have to do is we have to replicate it, right? So we can technically call, in this stage, it's still a chromosome. It's an unreplicated chromosome. When we get to this point, we have a replicated chromosome made up of 2 sister chromatids. Okay? So when we look at this structure, all 3 of these can be considered a chromosome. This is an unreplicated chromosome. These 2 are replicated chromosomes. Here, the only difference between these 2 is this one is packaged up a little bit more densely than this one. Okay? Notice the red dot on all 3 of these is where those It's the center of a single chromosome, but it's also where the sister chromatids are attached together, and that region is called the centromere. Okay, so now let's get to the whole meat of this cycle. When we think about the cell cycle, as I mentioned, it has two main phases. One is interphase, and the other one is the mitotic phase. The mitotic phase is where cell division is actually occurring. When we talk about interphase, interphase is the portion of the cell cycle where the cell is preparing to divide. This includes growth of the cell as well as replication of the DNA and preparing for that whole division. When we think about the mitotic phase basically has two different parts. You have the mitotic part, which is basically division of the nucleus itself. So when you are done with mitosis, you have one cell with two nuclei. You've separated out the nuclear material. And then you have to divide out the cell. And that's what cytokinesis is. It's division of the cytoplasm. If we look a little bit closer at interphase itself, interphase can be described as the portion of the cell cycle where the cell is most likely to be found. In fact, 90% of the cell cycle is in this portion, in interphase. Right? So if we think about this cell cycle again, this whole portion right here, this more than 3/4, because the mitotic phase is so small, is in interphase. And interphase is broken up into three different subphases: G1, S, and G2. G stands for gap. That's what the name is referring to. But what I want you to think about it as is the G is for growth, because that's mainly what is happening in the G phases, right? So in the G1 phase, this is right after mitosis has completed. So the cell has just been born and it's small. And so in the G1 phase, it's starting to grow and get to its proper functioning size. Once we get into the S phase, S stands for synthesis. And what we're doing is we're synthesizing the DNA. This is where DNA replication occurs. After we have replicated our DNA, we go back into another growth phase. Cell gets a little bit larger, but at this point, the other thing that we're doing is we're getting ready to divide. And if we're going to be producing new daughter cells, we need to make sure that each of those daughter cells has all of the organelles and everything necessary to be able to function. So this is also where we start to replicate organelles and that sort of thing. Okay. So in all three of those phases of interphase, the cell is growing, but only in the S phase does DNA replication occur. So the other part of the cell cycle is mitosis. And again, mitosis is division of the nuclear material. Mitosis is basically broken up into 5 different phases. We have prophase, prometaphase, metaphase, anaphase, and telophase. So if we think about these, we break them up into these different stages because different things are occurring, but it's more like a blended. It goes from one into the next. And some you can't even distinguish when you're looking at them under the microscope if we didn't know what was actually going on in those phases. Okay. When we get to telophase, at the end of telophase, cytokinesis has already started before telophase is finished. And so before we go into those different phases of mitosis, I want to talk a little bit about some of the other things that are really important that allow for mitosis to occur. And those are things like the centrosomes, the mitotic spindle, the cohesins, and the kinetochore. Okay, so when we think about the centrosomes are really important. These are basically microtubule organizing centers. These are the portion in the cell that produce the apparatus or the machinery that allows for that tug of war between the two different copies of the DNA and allows for them to be pulled apart. Right, so our centrosomes are composed of 2 different centrioles, and it's out of these centrioles that we start to have our spindle fibers coming out. And out of those centrioles, we produce our mitotic spindle. Right, so our mitotic spindle in this image is all of this green stuff here. So here is my centrosome made up of 2 centrioles that is producing those mitotic spindles. On the backside, you can kind of see it on these. At the backside are the asters. Okay. In this picture, the center portion, that's kind of the blue right there, that is the chromosomes. Okay. So when we think about when this is happening, prophase, that very first step in mitosis, is when we start to assemble this spindle, right? And it's occurring in the centrosome portion. The other thing that is really important are our cohesins and kinetochores. So remember when I said you have your chromosome, and then you replicate your chromosome, and your chromosome is then made up of 2 chromatids, right? So if this is my replicated chromosome right here, each one of these would be a sister chromatid. They're carbon copies because all we did was copy our DNA over. This region right here is the centromere region. That is part of what kind of holds those chromosomes together. In addition, you can see these little purple lines right here. These are proteins that are basically structures that keep the sister chromatids together. Those are cohesins. They cohere the 2 sister chromatids together. The other important thing, region that I want to point out is this region right here. This is the kinetochore region of the centromere. This is where those mitotic spindles attach, right? So if we think about this overall picture, here is my centrosome made up of 2 centrioles that's producing the spindle fibers that is attaching at the kinetochore region of the centromere of the 2 sister chromatids.